Okay, so the nucleus again is an organelle that we only find in eukaryotes. It's got two cell membrane or two plasma membranes around it, and in between those two membranes you've got holes or pores inside so that things can go inside and outside of the nucleus. Now, inside of the nucleus, what you find is something that they call chromatin, which is another name for chromosomes, and they're chromosomes when they're not dividing. So when you look at it under the microscope, it looks different, so they gave it a different name, chromatin, even though really what it is loosey-goosey, uncondensed chromosomes. And what it's made up of is protein. That's the thing that helps pack down, turn on and turn off the genes, and the DNA. And the DNA is what is actually, some of it is genes, some of it is material in between the genes, but it is what's carrying the instructions for the cell, how to make proteins. That is a key point. You should really know what the DNA is making instructions for, and the key is it's making instructions on how to make proteins. We'll talk a little bit later about why that's so important, but here, just to scratch the surface, it's important because proteins are enzymes that do the jobs in our bodies, and if we don't have the proteins that are made the right way, they're not going to function the right way, and then ultimately your body's not going to function the right way. The cells aren't going to function the right way. So it's very important that the DNA carries the correct information. Okay, this is a picture of the nucleus from one of your books, the book that I'm using, and you can notice the light purple, that is the nucleus. It's got two membranes, and you can see the little holes that go through into the nucleus itself. The chromatin is inside. You can tell it's kind of diffuse, and there's no like real chromosomes, which we'll see later when we talk about cell division, but that's what it looks like when the cell's active and working and making lots of proteins and doing the cell jobs. It looks like chromatin. It looks like not much of anything. And then they also have a picture in there of nucleolus. That's a specialized structure that just makes the RNA for the cell, and we'll talk about that a little bit later when we talk about the function of DNA. But for now, it's a little bit darker than the rest of the nucleus. And we'll talk about those dark purple structures in just a minute, but as a preview, they are the endoplasmic reticulum, and all the little bluey dotty things are the ribosomes, which are very important in making protein. Okay, so I just talked about the ribosomes, the little blue dotty things that we were looking at, and they are the structures that carry out protein synthesis. So the information is coming from the nucleus and going to the ribosomes, and the information is coming out as RNA, just so you know. So these ribosomes, you can find them in different places. You can find them, like we saw in the picture, attached to the endoplasmic reticulum, or you can find them floating around, just freewheeling in the cytoplasm, not attached to anything. The difference between these two types of ribosomes is what kind of proteins they make. If you find the free agent ribosomes floating in the cytoplasm, those tend to be making proteins that are going to work for the cell, do jobs for the cell. And if you find the ribosomes that are attached to the endoplasmic reticulum, those ribosomes are going to be making proteins that are for export from the cell. So things like insulin, which your body dumps into your bloodstream so that after you eat a donut, so that your cells can use glucose. Without the insulin in your bloodstream that your cells are making, you would be in dire straits. So anyway, the ribosomes that are doing this are on the endoplasmic reticulum. Now, to throw a further loop in endoplasmic reticulum is there's actually two different kinds of endoplasmic reticulum. I'm going to call it ER from now on because I'm going to trip over my tongue if I don't. Anyhow, the rough one is the one that we've been talking about, the rough ER. It has the ribosomes attached. That's why they call it rough, because it looks rough. It actually is connected to the nuclear envelope because it's exchanging information with the nucleus. And as we've talked about, it is primarily responsible for making new proteins, and particularly proteins that the cell is going to be exporting from the cell. Now, the way that it's done, and we're going to talk about it in a minute, is that the proteins are packaged in these little things called vesicles, which are just membrane with proteins inside, transported to another organelle called the Golgi, and it will be then exported from the cell. Now, the other type of ER is the smooth ER. So it's missing, you guessed it, the endoplasmic reticulum. The ribosomes. The smooth ER does not make proteins because it doesn't have the ribosomes. It makes lipids or fats. So, and again, anything with ER is going to be for transport outside of the cell. So these are fats that the cells are going to export from the cell. A really good example of this is steroid hormones, things like estrogen and testosterone are made in smooth ER for export, again, for specific cells. So those are cells that are going to either be found in ovaries or testes. They wouldn't be found in a brain cell. And again, this is a picture of the rough ER, the smooth ER. You can see on the rough ER all of the little dots, which are the ribosomes making proteins. And you can also see they've left a little bit of the nuclear envelope on there at the top part of the screen. In light purple, so they show you that the rough ER is connected to the envelope nucleus. And the smooth ER down on your lower right hand side is where the lipid synthesis is taking place. Like I said, you can't see the ribosomes because there aren't any. And if you look down below in the electron microscope picture, you can actually see a cross section, so you can see the smooth ER, which actually, to me, it kind of looks like little fat droplets, versus the rough ER, which has little bumpy things all over it. So those are pictures of them. The other thing I want to mention at this point is, and I kind of did before, is that certain cells are going to have more of some of these organelles than others. So if you have a cell that's transporting a lot of testosterone, like it's in the testes, you're going to find a lot of smooth ER in that cell. Whereas if you find a cell that's not doing so much transporting of lipids, like a muscle cell, but it may be doing a lot of transporting of proteins, you'll find more rough ER than smooth ER. So the cell can actually modify how much of these things it has, depending on what function the cell has. Now, before we finish up on the rough ER and what it's doing, I want to show you this picture of how those little vesicles actually work. So if you notice, again, in the left-hand side of your picture, the ribosome next to number 1, that ribosome is making a protein. It's putting it inside the rough ER, the membrane. And the membrane will actually bud off, if you see number 3, with the protein inside and make this little thing they call a vesicle. The vesicles transport the protein around the cell, and eventually it's going to be repackaged and sent out of the cell through an organelle called the Golgi. So this is how the rough ER works. This is why, remember, we said that This is the part of the cell that's making proteins for export. These little vesicles is how they do it. Okay, so we just talked about the vesicles. These are really small organelles and very, very simple. They're just a membrane, which is just like the cell membrane that we talked about. And there's stuff inside for transport. So proteins we just talked about, that could be lipids that we talked about briefly, just something that's going to be transported either around inside the cell. It can go from one part of the cell to another, or export outside of the cell. And we're going to talk about the ones that are going to be transported outside of the cell that have come from the ER. Okay, so once the vesicle is almost ready to get transported outside of the cell, they need to go through this thing called the Golgi complex. I like to think of the Golgi complex as the Homeland Security people who check your bags at the airport, because essentially they're doing the same kinds of things. The Golgi complex will take in proteins, they repackage it, they may take things out of it, they may add things into it. Like I said, just like people checking your bags at the airport. If you've ever had something missing from your bag, sometimes that's what happens in the Golgi complex. And then it will get repackaged and it will be exported outside of the cell. So what they look like, they just look like flattened membrane-bound sacs stacked on top of each other. Like I said, they're receiving internal vesicles from one side, they repackage in between, and then they export from the cell on the other side. Not only are they repackaging and sending things outside of the cell, but that membrane around the vesicle actually acts to replace the plasma membrane. So if the plasma membrane gets damaged in any way, or the plasma membrane takes something in and it gets smaller because it makes its own vesicle, that's how the cell replaces its membrane, is by exporting things using the Golgi complex. And again, from the book, this is a picture of the Golgi and vesicles. If you look at the artist rendering up in the top left-hand corner, you see the little purple transport vesicle from the ER. You see the orange Golgi, so it kind of hits it and becomes one with the Golgi, gets repackaged, and in the lower right-hand corner you can see the little vesicles coming off of the Golgi, and they will merge with the yellow, which is the outside membrane of the cell. If you look to the far right, you can see the electron microscope picture, and at the top is the Golgi apparatus, and it's just that little stacks and stacks of membranes. So that is the Golgi and vesicles, and we will talk a little bit about breakdown in just a second. Okay, so sometimes the cell will also take in food, and the way it breaks it down is through an organelle called the lysosome, which is mostly composed of enzymes, which we've talked about, that break down proteins, sugars, fats, harmful bacteria, and broken down organelles as well. The breakdown of these products can be used to make new proteins for the cells, new sugars, new fats, essentially breaking it down into its component parts so that the cell can use the building blocks again to make its own products. And again, a picture of lysosomes and how they work. On the top, this is a lysosome that would be digesting food. So the cell has some food that is coming into it. It makes a little vesicle or a vacuole around it. So the membrane is around it, the food's inside. The lysosome, with all of its digestive enzymes, merges and digests whatever's inside of that vacuole. If you look down on the bottom, there's something called autophagy, and this is when the cell has damaged organelles and the lysosome actually breaks down the damaged organelles so that it can kind of clean up and recycle its own organelles.